Welcome to PK's classes. Today we'll study about the Dideoxy Chain Terminator method or Sanger method of gene sequencing or DNA sequencing. So there are two methods of DNA sequencing. One is the chain terminator method of Sanger, commonly known as the Dideoxy Chain Terminator method of Sanger, and the other is the Maxam and Gilbert chemical method of gene sequencing. So today we will discuss the first method, the Dideoxy Chain Terminator method or Sanger method of gene sequencing. So before that, let us see the DNA structure. So DNA is a double helix structure. You can see here the double helix structure, and there are two strands, and each strand is a polynucleotide chain of sugar phosphate. So sugar phosphate, and from each sugar, so here you can see the structure, sugar phosphate, the chain is there, and with each sugar there is a base, and the bases can be adenine, cytosine, guanine and thymine, and the complementarity between the bases is adenine with thymine A, T and double bond, and cytosine and guanine C, G, triple bond. So this chain is a sugar phosphate, like this, and to each sugar there is a base, and the base may be out of 4. Then the other strand base pairs with this base, and then this chain occurs, this double helical structure occurs. And if you see the structure of deoxynucleotide, and we can see here 1, 2, 3, 4 and 5. So in the 3 position there is the hydroxy position, and because of this the chain elongates. So this is the sugar, and deoxyribose sugar, then the base is connected here, and there is phosphate, and during the linkage this diphosphate bond is taken out, and it connects with the 3 hydroxy group of another sugar. And if you see under this, you can see here numbers are there, and this third position hydroxy here is connected to the phosphate, okay, of the 5th position of the next one. Then this third position is connected to the 5th position, and this is elongated in the 5-3- direction, and we get this DNA. So what happens, the principle of this gene sequencing, the gene sequencing means the sequence of the base. The sequence of the base we can know, and how we can know. The method here is the chain terminator method, where we use the dideoxynucleotide. So here you can see in the deoxynucleotide there is this hydroxy group which is absent here. So in the deoxynucleotide, this position there is no hydroxy in the second position. In dideoxynucleotide, the second position as well as the third position, both are not having the hydroxy group. So because of lack of hydroxy group in this position 3, so there cannot be elongation, and the phosphate cannot be linked, so there will be termination of the DNA chain elongation. So termination can be there. So now, based on this principle, we can use the dideoxynucleotide, and we'll terminate the chain, and we'll get a number of fragments of DNA. Then we'll run it through gel electrophoresis, and then we can analyze them to know the sequence of the bases in the DNA. So the PCR technique we use, denaturation, in which the double strand is converted to the single strand, then renaturation and annealing, then there we will do the pairing. We will do the pairing here. So the primer we will give, so that there will be pairing, and then there will be elongation, and that will result in, that is called synthesis. So for that we need to give the primer, the DNA template, the DNA polymerase enzyme which will cause the polymerization, and the deoxynucleotide triphosphates. The deoxynucleotide triphosphates of all the 4 bases, and that, as we have already studied in PCR, please go through that PCR video, and because of the presence of this reaction mixture, there will be elongation of the DNA template. And if we'll add fluorescently labeled dideoxynucleotide triphosphates, then all the 4 bases, then these are the 4 bases: dideoxy adenosine triphosphate, dideoxythymidine triphosphate, dideoxycytosine triphosphate, and dideoxyguanosine triphosphate. Then occasionally they will, as compared to the concentration of the deoxynucleotide triphosphates, we will have less concentration of this dideoxynucleotide. So occasionally they will bind, and when they will bind, there will be termination of the chain. So we will get a number of fragments of the DNA chains because of the termination by the presence of these dideoxynucleotides. So when there will be chain termination, and then we will get a number of fragments, and those fragments will run through the gel electrophoresis that we have shown here, the gel electrophoresis, and the larger size will remain at the bottom, and the smaller size the DNA will move upwards, and then we can analyze them. So gel electrophoresis and fluorescence detection followed by computational sequence analysis. So you can see here now. So this was developed by Sanger. So according to his name, it is also known as Sanger method, where the single-stranded DNA with unknown sequence and the DNA polymerase and the deoxynucleotides and the radioactively labeled primer, all these are now present to this in 4 separate sample containers. We have added in one dideoxy adenosine triphosphate, in other one dideoxycytidine triphosphate, in the third one dideoxythymidine triphosphate, and in the fourth one dideoxyguanosine triphosphate. Then we'll allow the DNA extension followed as well as termination, and after that all those 4 mixtures will be put in separate columns in the gel electrophoresis, and based on their size the longer fragments will remain at the bottom, whereas the shorter fragments will move forward. Then from this we will read the sequence in the new strand. So you can see here, so this is dideoxy guanosine triphosphate, so here G, so we'll write G. Then the next one is here, second one, and this is A, so we'll write A. Then the third one is C, so here is C. The fourth one is T, so here is T, and accordingly then the next one here, that is G, so here is G. That means if this is the dideoxy bases, then the sequence in the template will be the complementary. So complementary of G is C, complementary A is T, complementary C is G, complementary T is A. So this is now the sequence of the DNA template which we have taken initially. So in this way we can know the sequence of the bases in the DNA, and we can call that as the DNA sequencing process. So let us summarize. So the first step will be PCR with fluorescent chain terminating dideoxynucleotide triphosphates. Then the second step will be the size separation by gel electrophoresis, and then third step will be laser excitation and detection by sequencing the cell. So this is all about the dideoxy chain terminator method. In the next class we will study about the chemical method of sequencing, gene sequencing. Thank you.